Assalamu alaikum grade 12 students. Today I will explain document 2 of chapter 3 mutations and multiple alleles. In the last video you have learned that mutations in a gene lead to the appearance of many allelic versions of the same gene. This gene is called polyallelic gene, which will produce new phenotypes from an already existing one and thus will cause polymorphism. It is because of all the mutations that has occurred to the human genome, we are so genetically different and every one of us is genetically unique. You have also learned that a mutation is a spontaneous accident that changes the sequence of DNA. A mutation might lead to a change in the protein coded by this DNA sequence. I said might lead because actually not all mutations will lead to a change in the coded protein. By the end of this document, you will be able to learn about the different types of mutation and list their different effects, and you will study the multiple alleles of the gene coding for a blood group as an example of a polyallelic gene created by mutation. Mutations are classified according to the number of nucleotides affected by the mutation. A mutation that affects only one single nucleotide is called a point mutation, while the mutation that affects many nucleotides is called a stretched mutation. In this example, you can see that the sequence ATAG is deleted by the mutation. Mutations that you will study in this chapter are point mutations, and document B classifies point mutations into 3 types according to what change occurred at the DNA level. This document also lists the 4 different effects that these mutations have on the protein. Let's start by the different types of point mutations. To identify the type of a mutation, you need to compare the normal and mutated sequences of DNA. Here is the coding strand of a normal gene. A mutation occurs to this gene. It becomes a mutated gene. Notice that during this mutation, one nucleotide T is replaced by another nucleotide C. This mutation is called a mutation by substitution. Don't forget to mention that this mutation is a point mutation since it affects only one nucleotide. Let's consider another type of mutation that occurs to this gene to become a mutated gene. Notice that during this mutation, one nucleotide T is deleted. This mutation is called point mutation by deletion. Now, in this last type of mutation, notice that one nucleotide C is added to the sequence. It is inserted between the nucleotides C and T. This mutation is called a point mutation by insertion. So, the 3 different types of point mutations are substitution, deletion, insertion. Remember that a DNA will be transcribed into an mRNA which will later be translated to a protein. And if the DNA is altered or changed by mutation, the mRNA will definitely change. But will this change in the mRNA always lead to a change in the amino acid sequence of the proteins? According to the change that may affect the amino acid sequence, there are different effects for mutation. If this DNA is subjected to a point mutation by substitution, not only the DNA will change, but the mRNA will change as well. Originally, the mRNA codon CCC codes for the amino acid proline, but after mutation, the mRNA codon, which becomes ACC, codes for the amino acid threonine. Then we can say that the protein coded by the mutated gene is altered. This mutation is a missense mutation since it alters the amino acid sequence with one amino acid only. However, in some cases, the mutation can change both the DNA and the mRNA, but the protein is not altered at all. This happens when both the original and the mutated mRNA codon code for the same amino acid. Thus, the protein coded by the mutated gene is exactly identical to the normal one. This is a silent mutation since it causes no detectable change in the amino acid sequence of the protein. In this example, both the original mRNA codon GUA and the mutant codon GUU code for the same amino acid value. Silent mutations happen thanks to the redundant mRNA codons. Redundant codons are mRNA codons with different sequences but code for the same amino acid. You can identify many redundant codons in the table of genetic code. Now, sometimes, and due to a certain mutation, a stop codon might appear. As you know, normally a stop codon terminates the process of protein synthesis, disassembles the ribosomal subunits, and releases the sequence of amino acids formed. In this example, the stop codon UAG appears early in the sequence because of a mutation. Translation stops and the released protein will be truncated, which means incomplete yet. This is a nonsense mutation that results in a non-functional protein. If the DNA is subjected to a point mutation by deletion or insertion, not only one nucleotide will be affected. Actually, the whole reading frame of the DNA will be shifted after the site of a mutation, as you can see in this example. Since all the mRNA codons after the mutation are altered, then the whole amino acid sequence will be altered as well. This mutation is a frame shift mutation since it alters many amino acids in the protein. To better understand the frame shift mutation, consider this sentence. It is a meaningful sentence, right? Let's insert D between I and G in the word big. Now, what happens to the sentence? Is not the whole reading frame shifted one letter forward and thus all the words are different? Now, this is exactly what happens in frame shift mutations, where all the codons after the site of mutation are modified. So, these are the different effects of point mutations: frame shift, silent, missense, and nonsense mutations. To understand more how mutations affect our phenotype by affecting the coded protein, let's take the blood group as an example of genetic polymorphism. You all know that the blood group is determined by agglutinogens, which are glycoprotein molecules found on the surface of red blood cells. The agglutinogen on the surface of red blood cell is composed of two molecules, a substance H and a sugar. The type of sugar added to substance H of the agglutinogen determines its type. If there is no sugar at all, then the blood agglutinogen is O. If the sugar added is galactose, then the blood agglutinogen is B. And if the sugar added is an acetylgalactosamine, then the blood agglutinogen is A. Now, the type of sugar added to substance H depends on an enzyme called transferase. Transferase is a functional protein coded by a specific gene and synthesized by the cell. It is the gene coding for transferase that was mutated by different mutations to give different forms of transferase enzyme. Each enzyme adds a different sugar or doesn't add a sugar at all. Transferase enzyme was coded by three allelic genes A, B, and O that control the type of fixed sugar on red blood cells and allows the appearance of different blood groups. A to B or from B to A, there are 4 mutations by substitution that lead to the modification in 4 amino acids in the enzyme and change its function. Allele O is a result of a small deletion which codes for inactivated enzyme that cannot add any sugar to substance H. Actually, the gene for transferase is not the only polymorphic gene that we have in our human genome. Other examples of polymorphic genes created by mutations are HLA genes and beta globin gene that you can read about in document 3 of this chapter.